Hello everyone, welcome to this module on which we are going to discuss about heterospory, homospory and seed habit, and finally we will be talking about some stellar evolution. So firstly, we will be talking about homospory, basic definitions of homospory, heterospory and seed habit. What is homospory? Homospory is a condition in which isospores or similar types of spores are produced. In case of heterospory, the spores produced are of dissimilar shapes, that is, one is smaller and the other one is larger. Finally, the seed habit. Seed habit is termed as an event which occurred after the heterospory. That heterospory culminated into the seed habit. So, here we have a figure where we can see that from homosporous condition how heterospory had evolved and how we have this modern seed plants. So here we see that a homosporous life cycle in which the sporophyte is producing the isospores. The isospores on further germination is producing the gametophyte and finally the zygote is produced. So example is Lycopodium. From homosporous condition, a second event took place known as incipient heterospory. It is regarded as an intermediate between the homosporous condition and an heterosporous condition. In this incipient heterospory, the female strobiles or this female prothalli, if remains unfertilized, it acted as a male prothalli. This incident is seen in equisetum. Finally, we come to a condition known as heterospory or complete heterospory. But this event did not occur at a single event. A series of processes occurred. Firstly, the difference in size was very little. That is, the microspores and megaspores cannot be differentiated so much with respect to size. But slowly, this size difference was seen. Now, previously, as we see cholera, there we find that the spores were the microspores was 30 micrometer and the megaspores were 50 to 150 micrometer in diameter. But slowly, that difference became enlarged. Now, previously, the microspores were produced in the microsporangium, or the sporangium was the same for both microspores and the megaspores. But later on, the sporangium became differentiated, that is, the microspores were produced in the microsporangium and the megaspores were produced in the megasporangium. The microspores are usually larger in numbers and the megaspores are smaller in respect to the microspores. So here is the life cycle pattern of an heterosporous condition. Here you can see that the microspore is being terminated into an male gametophyte and a megaspore to an female gametophyte. So this type is seen in Selaginella, etc. So we can summarize all these events by saying that firstly there was an increase or decrease in the number of the spores. Secondly, previously in homosporous condition we saw that it was a monoecious gametophyte, but here we are seeing that it has become to be a dioecious gametophyte. There is a transition from an exosporic condition to an endosporic condition. This event has certain characteristics, that is, heterospory is regarded as the advanced form. Firstly, the gametophyte is getting its nutrition, that is endosporic nutrition. It did not have to depend on external medium for its food. Now this is the complete life cycle of Selaginella, which is regarded to have a complete heterosporous type of reproduction. Here we can see that how this had taken place. These are some of the examples of the pteridophytes that shows complete heterospory. The first one is Azolla, second one is Marcellia, Selaginella and Iosoetes. Second, we will be talking about seed habit. So seed habit is not an independent event and it is just an prolongation of the heterospory. That is, from homosporous, heterospory was formed and now heterospory will finally terminated into seeds. Now there were certain stages that is taking place in a seed habit. Firstly, there will be a development of the pollen capture. Secondly, development of the ovules from a pre-ovule condition, formation of the integument or origin of the integument, and hydrospermum type of reproduction would be seen. So how this development of ovule took place? Firstly, there were 4 megaspores. Now amongst these 4 megaspores, 3 degenerated and 1 was functional. This functional megaspore remained in the megasporangium until the embryo was formed. So, it was a formation of an endosporic megagametophyte within an indehiscent megasporangium, and ultimately there was an elaboration of the apical region of the megasporangium, and also there was a formation of the pollen tube. Now, in this figure what we see, firstly it was an homosporous ancestor and the spores are produced in this sporangium. Now, we saw in heterospory that there was a difference in the types of spores. One was acting as the microspores and the others as the megaspores. Here, as I told, that a single megaspore got enlarged and the others were aborted. Finally, this megaspore was surrounded by an integument and there is an apical region or the micropilar region which acted for the reception of the pollens. Now, this origin or formation of the integument can be visualized according to the fossil study, or through fossil study we can say about the origin of integuments. Various theories have been put up about this. According to some scientists, this evolution of integument took place through a cup-like structure called indusium. According to some, it may be from the frond-like structure. But there were two most accepted theories. One was the telome theory as proposed by Zimmermann, and the second was nucellar modification theory as proposed by Andrews. In the first figure, what we can see, these are the fertile telomes and these are the sterile telomes. The fertile telome had a terminal sporangia. Due to certain processes, one of these telome that have this sporangia got enlarged in size. This is present in the middle. The other sterile telomes that are present slowly surrounds this particular sporangia. Now, these telomes which are present, they slowly got fused and ultimately this integument was produced which enclosed the middle sporangia. This is the second theory, that is the nucellar modification theory. Here what we see that we have two megaspores. Amongst these, one was aborted and the other remained functional. The functional spore slowly got enlarged in size and it sank in the lower or distal position of the megasporangium. Now, this megaspores stayed there and the vascular strand, there is a disintegration or the elaboration of the vascular strands, and slowly the integument was formed, leaving the micropillar region. Now, talking about the evolution of pollen capture method. So, the pollen capture method is at the distal end of the nucellus, and in certain early seed plants we see that there is a hollow pollen chambers. After this, there is also an evolution from a pre-ovule to a perfect ovule. By pre-ovule, we mean to say that an ovule in which the integument is not properly developed, and with time when the integument is completely developing, we call it a perfect ovule. This is also been explained with the help of the telome theory. And ultimately, we come to this hydrospermum reproduction in which the pollen capture method is elaborated at the distal end and it has the pollen which has a trilate suture. And this is the complete structure of the seed where we have this micropillar end, the integuments, the pollen chambers and the archegonia containing the egg cells. So, this seed habit is just a show of the parental care firstly, and secondly the seed can stay in the adverse condition and can grow only when the conditions are favorable for growth. With this, we complete this event of Heterosporous to the seed habit. Now we will be talking about another evolutionary event, that is the stellar evolution. So what is a stele? So stele is basically the central vascular bundle that is present in the stem and the roots. So the stele comprises of the xylem and the phloem. So the xylem is present in the central core, it is being surrounded by the phloem. Now before going into the evolutionary theories or the evolutionary part, we need to know the different types of stele. The first or the primitive one is termed the protostele and the advanced one is known as the siphonostele. In protostele, we have three types of steles. One is the haplostele, one is the actinostele, and finally the plectostele. So what is an haplostele? The haplostele has a central xylem core which is being surrounded by the phloem. For actinostele, here we see that the xylem had become armed and in between them we have the phloem. And ultimately in plectostele, it exhibits lobe-like structures or ray-like structures and phloem occurs in between these plates. And these plates are all interconnected to each other. After protostele, we come to the siphonostele. A siphonostele usually occurs in vascular cryptograms where there is a presence of a pith. So the basic difference between the protostele and siphonostele is the presence of this parenchymatous pith. Now this siphonostele is, as the protostele, it has a xylem and the phloem, but at the center it has a small pith. Now this siphonostele can be amphipholic or ectopholic. In case of ectopholic, we have a single layer of phloem surrounding the xylem, but in case of amphiphloic, both inner and outer phloem is present surrounding the xylem. Now this siphonostele can further be classified based on leaf gaps. So first one is a solenostele or amphipholic siphonostele, in which phloem is present on both outside and inside of the xylems and there is a single leaf trace or single break. After solenostele, we come to eustele, which contains the vascular bundles are being separated by leaf stresses, leaf gaps or branch gaps, and it is not possible to differentiate between the leaf gaps and the intrafascicular regions. Ultimately, we come to the dictyostele. Here we see that the siphonostele or the solenostele is further dissected by the leaf gaps and the leaf gaps are overlapping to each other. And lastly, we will be talking about atactostele, in which the complete vascular bundle is scattered in the ground tissue, that as single unit these vascular bundles are present. So after this, we come to this concept of this stellar evolution. So according to this, there are basic two theories. One is the intrastellar theory and the other one is the extrastellar theory. According to intrastellar theory, we can say that in the middle portion where there is the presence of the xylem, that is in the protostele, these tracheids are not all being formed from the procambial cells, and this pith, the middle one, has a combination of both tracheids and the parenchymatous cells, that is, it is a medullated protostele. So with that, after that the leaf traces are formed and ultimately it slowly culminated into the siphonostele, having a proper pith with the xylem and the phloem and the leaf stresses and the leaf caps. In case of extrastellar theory, it is said that this particular invasion of the parenchymatous tissues is occurring from the cortical cells, that is, it is not present inside the xylem. Now this parenchymatous tissues is coming from the cortical cells through the leaf gaps and it became trapped between the xylem. So the middle portion is the parenchymatous cells surrounded by the xylem and then the phloem. And as such, the leaf traces are formed before and slowly it goes on to form the siphonostele. So here in this diagram, what we see that firstly it's a complete protostele without any pith. Slowly here this pith is formed as the leaf traces are coming in, and there is the parenchymatous tissues, and slowly as such the complete a prominent pith is formed with the leaf traces. So here we can see this is a complete siphonostele with a C-shaped leaf trace. So here is then another figure, that is, here we will be talking about how this eustele is formed. We have seen that eustele, it is in dissected vascular bundles are present. So this was also formed from this specific protostele. So here you can see the protostele. So this protostele has a longitudinal divisions to form this medullated protostele, and finally this pith is coming in, and slowly the siphonous stele with formed with this many leaf gaps, and ultimately this terminated into an complete eustelic conditions. So this is how the complete siphonostele condition came into being from a very primitive protostele. So here we can conclude with that in the primitive plants we used to see a protostele. Now from protostele, three lines of evolutions have taken place. In one line we have these actinosteles, where we have seen that the xylem is star-shaped or armed and having phloem in between. From the actinostele, plectostele is formed, where the xylem has become more reduced, or it is present in plate forms which are interconnected with each other. When we will be talking about the other two lines, from protostele, the siphonostele was formed. From protostele's two lines, amphifolic and ectopholic. From amphifolic siphonostele with continuous overlapping of leaf gaps, this dictyostele was formed. So in the third line of evolution, we saw that there is an ectopholic siphonostele. From ectopholic siphonostele, the solenostele was formed with the leaf traces, and from solenostele ultimately eustele was formed, and finally from eustele this atactostele formed, in which the vascular bundles were scattered in the ground tissue. This atactostele is the most advanced type of stele, and it was seen in the present day monocot plants. So in this module, what have we learned? We have learned that this heterospory have originated from an homosporous conditions, that is, in the primitive plants we have only seen the homospores, that is isospores were produced, and to attain more reproductive success this isospory slowly got transferred, that is there was a difference in the sizes, and microspores and megaspores were formed. Now with time there was a difference in their sizes, that is the sizes becomes more elaborated. They were present in different types of sporangium rather than being present in the same sporangium. So microsporangium and megasporangium came into being. So there was a transfer from an monoecious conditions to an dioecious conditions, from exosporic to an endosporic one, and there is also in the reduction of some spores and the elaboration of the others. So nutrition also sometimes played some role here, because the microspores were there was little nutrition was given to this microspore, where megaspores had large of its share. So that's how this heterosporous, complete heterosporous condition came into being, while in the middle we have that intermediate stage of incipient heterospory. After this we talked about the seed habit, or the seeds that was considered as the most advanced formed. So here we have talked about that how these integuments were formed, how the ovules were formed from an pre-ovule structures, what theories were there, these Zimmermann's models, these Andrews models, which talked about the various ways in which slowly the integuments came into being, and ultimately the pollen capture method, how it got elaborated, and ultimately we saw a complete seed was produced with its proper integuments. And ultimately we talked about the stellar evolution, the various types of steles which started from the protostele and ultimately culminated into the siphonostele or siphonostele conditions, and as such we concluded that siphonostele was the most advanced type of stele, and we see this in present day angiosperm. Thank you.